Welcome to Unit 24. This is essentially part 2 of the lecture series. So in this chapter, we continue talking about the digestive system. This time, however, instead of just naming the organs and where they are located, we're actually going to talk about some of the histological components of the digestive system and some of the physiology involved in properly digesting the food that we've eaten. Now, essentially, when we talk about any kind of hollow organs, whether it's like we talked about the respiratory system or now digestive system, we always see about 3 layers when we do a cross section. We see the internal layer with the normal epithelium, that's the mucosa. We see the submucosa underneath, okay, submucosa like that, and then obviously the externalmost layer. In this case, it's going to be the muscle. These are the muscularis layers, internal muscularis layers. Other additional layers are possible, of course, but these are the primary histologic layers that we're going to see. So now I'm not going to go through every single organ in the digestive system to point out what kind of mucosa and other layers are there, but we're going to kind of hit upon the main points, the key points here, try to understand how this works. So primarily keep in mind that in the digestive system, the normal digestive epithelium, where absorption actually takes place, is actually the simple columnar epithelium, right? These are tall cells, columnar cells, 1 layer thick. This is the best kind of epithelium, most optimal for absorption of nutrients. But keep in mind, of course, throughout the digestive system, there are many areas, such as the esophagus or the oral cavity, other areas, where absorption doesn't actually take place, and so the food is just kind of in transit in that area. So they are mostly, again, in the oral cavity, in the esophagus, we see stratified squamous type epithelium. Now, what kind of other unique features do we have here? So for instance, in the esophagus, besides the normal stratified squamous epithelium, for the most part, we also keep in mind that because absorption does not take place there and the food is just passing quickly through that area, we do not have mucus secreting goblet cells, so no goblet cells. It's important to know this just to kind of keep this in mind when you're like looking at the histology slides. And when we go into the stomach now, we do see goblet cells, we do see lots of gastric glands, we do see all kinds of endocrine and neuroendocrine type cells there. And very unique feature also of the stomach is to know that there's a presence of this what's called mold or mucosa associated lymphoid tissue. This is specialized types of cells, essentially that bridge the lymphatic system, digestive system, and the immunological processes of the body, the immune system. And so we have a collection of specialized cells that protect essentially the stomach and protect the digestive system. Again, we call them mucosa-associated lymphoid tissue. Okay. Now, once we move on to the small intestine, again, small intestine, remember, is over 20 feet long. There's quite a lot of surface area increases in the small intestine to increase the chances for absorption to take place. So the main cell types in the small intestine are going to be the enterocytes. Again, these is still the epithelium is the same. It's simple columnar epithelium, but when you take the individual cells that are doing the absorption, we call these enterocytes. Again, entero, keep in mind, essentially always means deep inside. Here, obviously referring to the digestive system. And we have all kinds of intestinal folds there, and besides the general length of the small intestine. And additionally, these folds now form as villi and then microvilli, right, which are these like invaginations throughout the cells. And again, the goal ultimately here, whether it's at the microscopic level, like the microvilli, or those you can see by eyes with the others, is to increase surface area to maximize absorption of all nutrients, of all the different molecules. So that's the primary goal here, because remember, most of absorption, most of the breakdown and processes of digestion actually take place in the small intestine. Okay, so when you see this in illustrations, when you see this in the book and in histology slides, you're going to pay attention to these different components in the small intestine. Now, let's quickly move on to the submucosa. What's on the submucosa, right? That's the kind of essentially support tissue. We're going to have blood vessels there. We're going to have nerves. We're going to have other areas there, other components like part of connective tissue, just kind of to support the rest of the higher levels of that structure, those that lie above, like the mucosa. Okay. So again, areas of submucosa will vary, and different components will be different depending on where you are in the digestive system. Right, makes sense. And you'll see that sort of when you look at the illustration. So depending on the slides, you'll see different components there. When we look at the pancreas now, this is a solid organ now. Obviously, the pancreas also consists of different cells. Remember, we always separate pancreas histologically and functionally into exocrine pancreas and endocrine pancreas, even though it's one solid organ. That component that contains cells connected to major pancreatic duct and the minor pancreatic duct. This is the exocrine component. And the primary cells there that produce and secrete digestive enzymes and bicarbonate are the acinar cells, A-C-I-N-A-R, like that. That's how they're spelled. Acinar cells are essentially normal gland-like cells that deal with secretions. In this case, secretions going into ducts. Eventually will go into duodenum and help to mix in with the food to help break it down. Again, using all kinds of digestive enzymes. We're going to talk about them very soon here. The endocrine portion of the pancreas is the hormonally active portion. These are specialized cells. They're called islet cells or islets of Langerhans. These secrete hormones like glucagon, insulin, and some others. Okay, so insulin is secreted by the beta cells or beta cells that are endocrine, and alpha cells are designated to secrete glucagon. Again, keep in mind that insulin is essentially secreted in response to hyperglycemia. So after a meal, you had all kinds of carbohydrates now in your digestive system, and in order for the sugars, as they're being broken down, to finally be absorbed by cells, insulin must be present to accept glucose into the cells. Okay, we'll talk about more of this and how it relates to diabetes in the endocrine chapter. When we talk about the liver, again, the liver, very large, very complex organ, as we said before. It's a solid organ consisting of hepatocytes, but the hepatocytes are not arranged randomly there. There's a specific way that we can kind of organize these. There are multiple, actually, varieties of organization. The best one, the easiest one to talk about is the portal triad. This is essentially keeping in mind that bile needs to somehow be produced and be carried throughout the liver, and obviously we need the oxygenated blood to be supplied to hepatocytes, and also the hepatic portal system must somehow be present there as well. Remember, hepatic portal system refers to the blood that doesn't carry oxygen but carries actually the nutrients from the digestive system to be processed by the liver. So when we combine these three components, we have the bile duct, the portal vein, and the hepatic artery traveling essentially together in this portal triad. And between these components essentially throughout, you see the hepatocytes. Take a look, again, this is very visually kind of important area. Take a look in the book, in the pictures, what it looks like. There's different ways, again, to organize the liver. Sometimes liver lobules are used, sometimes other components. But overall, again, liver cells, hepatocytes are there, right? And they're kind of surrounding these portal triads. Bile duct, portal vein, and hepatic artery. The other components here, right, the last kind of external layer is the muscle layer. Right? Obviously, these are smooth muscles. We're talking about the involuntary muscles. They're often organs like the stomach and others that have many, many muscle layers. Just like we'll see, for instance, in the urinary tract, like in the bladder, and in some other organs. So muscularis externa, muscularis interna. These are layers that participate in peristalsis. That's the muscular contractions that occur as part of the digestive process, moving the food along to the next portion in the GI tract to allow for breakdown and absorption of the nutrients. Now, overall, also keep in mind that the largest of the organs in the body also often contain this thin external protective membrane called the serosa, and this is kind of similar to how we talked about the pleural membranes. For the lungs, right, we need these protective membranes, protected areas, and so the largest of the organs often in the digestive tract will have the serosa, again, just as a form of like a connective tissue protecting the organs. Now let's talk about physiology of digestion, essentially only focusing here on what kind of molecules, what kind of organic molecules will be broken down in the digestive tract and where will they be broken down. So three main organic molecules that need to be accepted by the digestive tract are the carbohydrates, monosaccharides, polysaccharides, basically sugars, the proteins, right, consisting of amino acids, very large molecules, a lot of variability there, often very complex in 3 dimensions, but eventually these proteins need to be broken down into their subunits, which are amino acids, and the fats or lipids. Right? So molecules like cholesterol, fatty acids, triglycerides, and others. For the carbohydrates, for the sugars, 2 main areas in the digestive tract where they will be broken down are oral cavities, so inside the mouth. We have salivary amylase. Okay, that's the enzyme inside the saliva that starts to break down of starch. And so again, salivary amylase, the enzyme starts to break down starch or carbohydrates in the oral cavity. And then most of carbohydrate digestion actually takes place inside the small intestine using what's called the brush border enzymes. These are the enzymes. They're called like that because the appearance of the villi and the microvilli creates this like brush-like appearance. And the enzymes secreted there, such as lactase, continue to break down sugars in this part of the digestive system. So lactase is there obviously to break down lactose or the milk sugar that we are familiar with. So people who are lactose intolerant have lower production of lactase or no production of lactase at all. And so after digesting some kind of a milk-based product, they will often have bloating, abdominal pain, sometimes diarrhea, things like that, right? So this kind of unpleasant effects because their body is unable to deal properly with the lactose in the milk due to the lack of the enzyme. Now, next important type of group of molecules are the proteins. Now again, as we said before, proteins need to ultimately be broken down into amino acids. But before we can do that, proteins need to be untangled, separated from their multiple different complex structures and interactions. And in order to unfold them, break them apart, and separate them into those final units, 2 main areas of the body do that. And one is the stomach, where pepsin, originally pepsinogen, must be activated by the hydrochloric acid, and then starts to essentially break down the protein. So pepsin in the stomach is responsible for protein breakdown, and that process continues in the small intestine with the help of at least 3 separate enzymes like trypsin, chymotrypsin, and carboxypeptidase. For the purposes of this discussion, the only thing you have to know basically is that trypsin and chymotrypsin actually originate from the pancreas, and once these pancreatic enzymes enter the small intestinal lumen, right, that area there, let's say in the duodenum, trypsin, chymotrypsin will be breaking down proteins. And then an additional enzyme to break down proteins is carboxypeptidase, but this one essentially originates as a brush border enzyme, kind of like lactase. Okay. For the final molecule, the lipids, fats, fatty acids, like triglycerides, cholesterol, these kind of molecules. Keep in mind that these are molecules that are very large, insoluble in blood, insoluble in water, so they are particularly difficult to deal with usually. Because of that, the body has instituted special kind of measures, more complex, stepwise procedures in order to help the body actually absorb these lipids eventually to break them down. So primarily this takes place in the small intestines when the gallbladder releases bile into the duodenum. The bile acids there start to mix in with the fats, and they actually create something called the chylomicron, which is a combination essentially of taking something that's insoluble and making it more soluble, and eventually this process allows these molecules to be eventually absorbed into the lacteals and the lymphatics, and eventually to the bloodstream. So again, emulsification by bile acids, right, is this important step in trying to turn something that's insoluble into more soluble components, and is an important part of lipid breakdown and digestion. And an enzyme from the pancreas called the lipase also helps with that process, also helps to break down fats in the body. Now, again, take a look at the rest of this chapter, look at all the illustrations, pay attention to the names of these enzymes, these different histological components. And when we look at histology, virtual histology, or basically histological slides, we need to review all of these areas and see how do they relate, how do they function together in the digestive tract.